principal excretory organ of the human body is a pair of kidneys, and the principal excretory product is urea. But urea is not produced in the kidney. It is produced exclusively in the liver because arginase, an important enzyme required for production of urea, is present only in the liver. Urea is produced by the urea cycle. The cycle is also known as the ornithine cycle, as ornithine is utilized as well as regenerated by this cycle. It is also called the Krebs-Henselt cycle. The cycle consumes ammonia, aspartate, carbon dioxide and ATP and generates urea and ornithine. One nitrogen atom of urea comes from ammonia and another nitrogen atom comes from aspartate. The first two steps occur in mitochondria and the rest steps take place in the cytosol. Let us start the journey of the urea cycle. In the first step, carbon dioxide and ammonium ion join together to form the carbamoyl phosphate. This step takes place in the mitochondria and is catalyzed by CPS1 or carbamoyl phosphate synthetase 1. This is the rate determining step of the urea cycle. NAG or N-acetyl glutamate is an allosteric activator of CPS1. This step utilizes two molecules of ATP. From two molecules of ATP, two molecules of ADP are generated. There is another enzyme known as CPS2 or carbamoyl phosphate synthetase 2, a cytosolic enzyme that is involved in the pyrimidine biosynthesis pathway. In the urea cycle, the carbamoyl phosphate then joins with ornithine to produce citrulline. This step is carried out by the enzyme OTC or ornithine transcarbamylase. This step occurs in the mitochondria, but ornithine is present in the cytosol. It enters inside the mitochondria with the help of an ornithine transporter. Then aspartate comes and joins with citrulline under the function of enzyme argininosuccinate synthetase, and argininosuccinate is produced. This step utilizes one molecule of ATP. Here, from one molecule of ATP, one molecule of AMP is produced. Then argininosuccinate lyase enzyme breaks the argininosuccinate molecule into one fumarate and one arginine molecule. Fumarate enters the TCA cycle and arginine is acted upon by arginase. Arginine gets converted to ornithine and as a byproduct, urea is generated. Ornithine then enters into the mitochondria and participates in the urea cycle again, and urea gets eliminated from our body. Let us see the energetics of the urea cycle. In the first step, two molecules of ATP are utilized, and in the third step, another molecule of ATP is used. In the third step, ATP gets converted to AMP. So, two ATP equivalents are used in the third step. Ultimately, the cycle uses 3 molecules of ATP and 4 ATP equivalents. Regulators of the urea cycle. NAG, the so-called sixth enzyme of the urea cycle, acts as a regulator. A high intake of dietary protein increases the rate of urea production. In case of starvation also, urea synthesis increases in our bodies because the enzyme level gets elevated to cope with the increased production of ammonia, which occurs due to starvation-induced degradation of protein. As I have said earlier, the reactions of the cycle are compartmentalized. Some occur in mitochondria and others in the cytosol. This type of compartmentalization also helps to regulate the cycle. Let us see some disorders associated with the urea cycle. Hyperammonemia type 1. It occurs due to defect in the enzyme CPS1 or carbamoyl phosphate synthetase 1 enzyme. Hyperammonemia type 2. It is due to a defect in OTC or ornithine transcarbamylase enzyme. Citrullinemia. There is a deficiency in the enzyme argininosuccinate synthetase. Next is argininosuccinic aciduria. It occurs due to deficiency of arginosuccinate lyase enzyme. Then there is hyperargininemia. Defect in arginase enzyme is responsible for this disorder. Lastly, there is a disease known as Triple H syndrome. This disease is characterized by hyperammonemia, hyperornithinemia, and homocitrullinuria. It occurs due to a defect in the ornithine transporter as a result of a defect in the ORNT1 gene. This is all for today. If you found this video helpful, hit the like button, share it, and do subscribe to my channel. Press the bell icon to get notified about new videos. Thank you and happy learning.